respiration and fermentation. And yeah, let's get started. Okay, so we're just going to be doing an overview, and then we'll talk about the specific steps, and then we'll talk about anaerobic respiration and fermentation at the end. Overview. Okay, so first we're starting off with glycolysis, and this happens in the cytoplasm of the cell. So we don't need oxygen for glycolysis to happen. It can happen in either condition. It doesn't need oxygen, but whether or not it has oxygen changes kind of where, the whole process is going. So if you have oxygen, you're going to go to the Krebs cycle, then to the electron transport chain, and make about 30 to 32 ATP. So this is what we call cellular respiration. Now, if you don't have oxygen, you're going to go down this path to fermentation, and you're going to make about 2 ATP, and this is per glucose. So, obviously you can see that respiration would be the better choice for maximizing the amount of energy you make, but some organisms, or when you're in a unsuitable environment, it makes it so you can't do respiration, and thus you can still get some ATP out of fermentation. Alright, so here are the energy sources that are being used in cellular respiration. So ATP, or adenosine triphosphate, is kind of the big one you hear talked about a lot in biology, and even the other energy sources shown here are going to get converted into ATP because it's just a really easy form of energy to access and use by, organisms. So ATP, we can get ATP with two methods. One of them is substrate level phosphorylation. This is when we gain ATP as a byproduct of something else that just happened. Or oxidative phosphorylation, which is when we get ATP from the electron transport chain. Okay, so phosphorylation is essentially how ATP can carry energy. So ATP, it's triphosphate because it has 3 phosphates on it, and then that last phosphate is kind of the important one. Essentially, wherever that phosphate is where the energy is. The energy is stored in the bonds between the phosphates. So if ATP is phosphorylated, the phosphorylated form is called ATP. That has energy. If something else is being phosphorylated by ATP, that thing has energy. And ADP is the kind of sort of deactivated form of ATP because it doesn't have that kind of energized phosphate. So the enzyme that kind of activates or deactivates ATP is called a kinase. Alright, so NAD plus and NADH stands for nicotinamide adenine dinucleotide. So, in a lot of ways, this is very similar to how ATP works. NAD plus is kind of like ADP, NADH is kind of like ATP. So, instead of carrying the energy in the phosphate, though, carries it in electrons. So NAD plus and FAD are both called electron carriers for that reason, because they sort of have the energy in that electron. So NAD plus, it's about worth 2.5 ATP in the end. Ideally it would be 3, but transporting things through the cell takes some of that energy that you made. And in order to make it, you need vitamin B3 or niacin. And yeah, so just the way that ATP had kinases to activate and deactivate it, NAD plus and NADH, it alternates between those two forms because of NADH dehydrogenase. Alright, so FAD and FADH2, largely the same concept, stands for flavin adenine dinucleotide. It's worth 1.5 ATP as opposed to 2.5, and it's made from riboflavin, which is B2. Yeah, and then the extra H you're seeing at the end for NADH and FADH2, that just kind of comes from the surroundings. It balances out the charge of the electrons. All right, so next is glycolysis. All right, so that big equation you see up top is the net equation for what happens in glycolysis, but basically you are breaking down one glucose molecule into two pyruvate molecules, which each have 3 carbons. So if you look at this chart, I've marked it up with some comments about different enzymes and different molecules. We're going to look at those last. Let's just walk through this chart first. So first we have our glucose, and then hexokinase, which is the first enzyme here. It takes 1 ATP and it phosphorylates glucose and turns it into glucose 6-phosphate. Phosphoglucose isomerase turns glucose 6-phosphate into its isomer, fructose 6-phosphate. So you haven't added anything, it's just you change the shape. And then phosphofructokinase takes another ATP, phosphorylates the first carbon here, turning this into fructose 1,6-bisphosphate. And then fructose bisphosphate aldose essentially cleaves this molecule in half, turning it into, this is just usually abbreviated as DHAP, and glyceraldehyde 3-phosphate, usually called G3P. And then this G3P is the form we want, and there's an enzyme triose phosphate isomerase that continually switches all of the DHAPs so they look like G3Ps and can go through the rest of this. So yeah, we're just making one of these, and then this gets turned into this anyways. So from now on, everything is happening twice for every glucose molecule. This is what's happening for one G3P. All right. And also, you know how we spent 2 ATPs here? We call that the energy investment phase, and now we're going to gain back some of those ATPs. So for each G3P, we take an NAD+, and we turn it into NADH. So if you'll see here, and we also add a inorganic phosphate. So we get 1,3-bisphosphoglycerate, and then we use phosphoglycerate kinase, and we actually make an ATP from one of these phosphates right here, and we get 3-phosphoglycerate, and phosphoglycerate mutase turns this into another form called 2-phosphoglycerate. And enolase, this is a pretty important enzyme because it makes water. Right here, it turns it into this form called PEP. And then pyruvate kinase finally turns PEP into pyruvate. So this happens for each G3P. So one glucose makes 2 pyruvate molecules. Now let's look at the commentary I put on the enzymes and molecules. So hexokinase, phosphorylating glucose, is what traps glucose in this cell. So glucose has a relatively okay time moving across cell membranes because there's usually mechanisms for it to. But if we phosphorylate our glucose, suddenly it has a negative charge that makes it much harder for it to leave the cell. So yeah, that's the importance of hexokinase. And then phosphofructokinase is important because it's a major point for regulating how much glycolysis happens. You don't want glycolysis happening all the time, especially when you don't need it. But you do need it to happen when you need it to happen, right? So if you think about it, we need glycolysis if we have too much ADP and not enough ATP. So the presence of ADP, this little arrow just means that it essentially positively regulates phosphofructokinase. So more ADP means phosphofructokinase has more activity. And ATP and citrate, so ATP, obviously if we have enough ATP, we don't need to be going through all of cellular respiration to make more. So the presence of ATP inhibits phosphofructokinase's activity, meaning that less glycolysis happens. And citrate is something that comes along later in the Krebs cycle, but essentially if you have a lot of citrate, that just means you're doing cellular respiration, you're doing a lot of it, you don't need more of it to happen. So yeah, these two would inhibit phosphofructokinase. Now we have fructose 1,6-bisphosphate. So this molecule, if we have a lot of it, it's going to increase the activity of pyruvate kinase. And the way this works is basically if we have a lot of this product from earlier in glycolysis, we're telling this enzyme to be more active because it needs to finish that process right here, right? So it basically is just kind of like a signal like, hey, we need more of you to do work later. So the products we get out of glycolysis are 2 ATPs and 2 NADHs and no FADH2s. So yeah, this is our total for glycolysis. Now the intermediate step. Okay, so here's a picture of what happens, but basically that pyruvate we just got out of glycolysis, it's still in the cytoplasm, the cytosol. It's not in the mitochondria yet, so we're going to get it into the mitochondria, where the rest of our cellular respiration is going to be happening. So pyruvate's going to come through a transport protein, and a CO2 comes off of it. So this is the first point in cellular respiration we're making CO2. If you, humans, we breathe out a lot of CO2. That's where all that CO2 is coming from. It's coming from cellular respiration. There's more later, but this is the first place we're getting CO2. We also make an NADH off of this pyruvate, and then we add something called coenzyme A, which is this group right here, and we end up with something called acetyl-CoA. And this is what's going into the Krebs cycle next. So what we got out of this, no ATPs, so we still have 4. We have 2 NADHs, so we have 4 NADHs now, and no FADH2s, so none yet. Alright, so now the Krebs cycle, or called the citric acid cycle, or the tricarboxylic acid cycle. So what we're putting in, if you remember, is that acetyl-CoA here. So what happens is that CoA gets taken off, and then citrate synthase makes it into a citrate, and then it goes through all of these steps right here. You can look at it more specifically. It's useful to know all the enzymes in the products here. You don't necessarily need to know what they look like though. But here is a mnemonic that I like using for remembering all the steps. There's not really much to it. It's just something I can say in my head, and it has a certain ring to it, so I can remember it. But yeah. So this is just for all the products. So C for citrate, A for cis-aconitate, and etc. I would say that the enzymes are pretty intuitive to remember. Aconitase makes aconitate. Isocitrate dehydrogenase, takes a hydrogen off of D-isocitrate, makes it alpha ketoglutarate. So yeah, generally if it's something dehydrogenase, that comes after that something. But if it's something like a synthetase or a synthase, that makes the thing that comes after it. So yeah, here we get 2 ATPs, we get 6 NADHs and 2 FADH2s. So the big idea of the Krebs cycle really is to strip off all of the carbons from our pyruvate and turn, those get turned into a bunch of CO2, which is kind of where a lot of the CO2 from, us breathing comes from. But yeah, just stripping all of those off and making as many, units of energy as we can to prepare for the electron transport chain, where we're going to milk all of this NADH and FADH2 for as many ATPs as we can get. Now we're on to the electron transport chain. So this is the last step of cellular respiration. It's where we're going to get the bulk of our ATP. So we're starting by stripping off the electrons from NADH and FADH2, a bunch of which we just got. And then the electron transport chain's proton pumps are going to use the energy from those electrons to create a proton gradient. So this gradient is going to be really important for the next step, which is called chemiosmosis, in which protons flow through ATP synthase, an enzyme that makes ATP, to make ATP. So yeah, that's how we're going to get a bunch of ATPs from NADH and FADH2. All right, so here is what that looks like. So we see this electron transport chain. This picture has all of these specific, enzymes inside of these complexes. But just know there's a complex 1, 2, 3, 4, and 5. Complex 5 is ATP synthase. And essentially, let's just walk through this whole thing. But first, know all of these complexes, the electrons that arrive at each of them get lower and lower in energy level because most of them are going to be pumping protons outside into the intermembrane space to create that gradient I was talking about. So yeah, let's just walk through it. So complex 1 is where NADH drops off its electrons. It's also called ubiquinone oxidoreductase, and those electrons, they get carried along with the electrons from FADH2, which are dropped off at complex 2, also succinate dehydrogenase, which is also present in the Krebs cycle. These get carried by what they call an electron carrier called Q, or ubiquinol. If you've ever seen those ads or supplements called where it's like CoQ10, this is the same stuff. And then it's important to note that Q is in the membrane, which means it's lipid soluble, it's hydrophobic. And then that carries the electrons through complex 3, also called this really big name here. Again, it's just pumping. Notice that complex 2 is the one that doesn't pump. And then that gets transferred to cytochrome C, which also has a function in apoptosis. It has a porphyrin ring or a heme group in it. Essentially, it just has iron, which helps a lot of its functions. But yeah, you can see that it's water soluble because it's outside of the membrane, and that's a very big, important difference to know about ubiquinol and cytochrome C. So cytochrome C takes the electrons to the last complex that pumps. So that's complex 4. Cytochrome C oxidase because it oxidizes cytochrome C by taking an electron from it. So again, these three are going to be pumping our hydrogens, our protons, outside of the matrix so they can all flow through ATP synthase by diffusion. So yeah, essentially we just milked all of the energy we could out of those electrons to pump a bunch of protons over there, and then all those protons are now going to come through complex 5 or ATP synthase. And what that's going to do is it's going to make a bunch of ATPs. We'll talk a little bit more about that, but first, what happens to all those electrons that lost, all their energy in the electron transport chain is they get added to oxygen, you can see right here, and that creates water. So that's why cellular respiration creates water, and that's why we need oxygen. Okay. So this is just some more specifics on how ATP synthase works. So first, what's going to happen is one of those protons is going to come in this place called the stator. It doesn't move. It's going to come through a little channel there. It's going to go on the rotor, and the rotor moves. Every time a proton is added, it moves a little bit, and that turns a rod, which turns a knob. And then that knob is what turns ADP and inorganic phosphates into ATP. So yeah, that's just how ATP synthase works. Essentially, it's just hydrogen ions moving in, and that creates ATP. So this is what we got out of cellular respiration in terms of ATP. From oxidative phosphorylation, which is another term for the electron transport chain process that involves essentially the electrons powering the phosphorylation of ATP. We got 34 ATP. We lost all of our NADHs and FADH2s. So this chart here just totals up all of that. You can take a good look at it. It's not too important to know all of the numbers. I would know them, though. Just they're not terribly hard to remember. I think it's a good thing to know still. But you're most likely never going to be asked, how many ATPs are made in, the Krebs cycle or something like that. But yeah, and you'll notice that there is a net total here with transport, because transporting everything throughout the cell does take energy a lot of the time. So we're going to end up in the very best case scenario with 36 ATPs. But, a lot of the time it's not so ideal. We're going to end up with maybe 30 or 32 instead. We're on to anaerobic respiration and fermentation. So these are kind of like variations on cellular respiration, under different circumstances. So anaerobic respiration is really, really similar to cellular respiration, but instead of using oxygen at the end to receive those electrons that are out of energy after being through the electron transport chain, you use different compounds. So sulfate, or SO4 2- is sometimes used, and that creates hydrogen sulfide, H2S, instead of water, H2O. So yeah, it's really, really similar, and this is a different thing than fermentation. So fermentation, the whole point of it really is to recycle NAD+ while getting as many ATPs off of it as you can. So there's two types: alcohol fermentation and lactic acid fermentation. So alcohol fermentation, it makes your alcohol. That's why you seal, like when people are making alcohol, they seal like the barrels and stuff, because the yeast, they need to be under a anaerobic condition to be able to ferment. And lactic acid fermentation, if you've ever been running and your legs really hurt after a while, it's because your breathing is not giving your legs enough oxygen to just be able to get energy off of cellular respiration, because that energy comes a little slower. So you're going to be using fermentation, and that's going to make lactic acid, which is what's making your muscles hurt. So yeah, if you look at each of these processes, essentially, you're just recycling NADH, like this, and then you're just using glycolysis, essentially, to try and get as many ATPs as you can. And then each cycle with each glucose only gets you 2 ATPs, so it's vastly unpreferred to cellular respiration, but some organisms only use fermentation, and under some circumstances you are forced to use fermentation. Alright, thank you so much. I really hope this video was helpful. If it was, please let me know by leaving a like or a comment, and please subscribe for more content.